Good evening, everyone, and welcome to the 21st episode of UCD in Conversation, a virtual series hosted by the UCD Alumni Relations team. My name is Professor Cecily Kelleher, and I'm the principal of UCD's College of Health and Agricultural Sciences, and it is my pleasure to introduce tonight's event. Through this series, we welcome our alumni community from around the world to listen to fellow alumni, UCD academics and guests share their stories and ideas. The series also reflects UCD's Rising to the Future Strategy 2020 to 2024, and its four thematic pillars have carried through the conversation topics: creating a sustainable global society, transforming through digital technology, building a healthy world, and empowering humanity. When we launched the strategy just a year ago, we could not have imagined how pertinent these themes would become to the pandemic crisis that was to unfold. COVID-19 has disrupted and taken lives across the globe since it first emerged at the end of 2019. We have seen an unprecedented public health global response and outstanding scientific endeavour to develop an effective vaccine strategy. This is the theme of tonight's panel discussion. It is my pleasure to introduce you to our distinguished panel. Our moderator for the evening will be Dr. Cillian de Gascun, who is consultant virologist and director of the National Virus Reference Laboratory at UCD, and chair of the SARS-CoV-2 Expert Advisory Group, a subgroup of NEPHIT, the National Public Health Emergency Team. Cillian will be speaking with and facilitating our two guests this evening. Professor Karina Butler is chair of the National Immunisation Advisory Committee, member of the high-level task force on COVID-19 vaccination, and consultant paediatrician and Professor of Clinical Pediatrics at UCD. UCD alumna, Professor Teresa Lambe, who worked previously at the UCD Conway Institute, joins us from the prestigious Jenner Institute at the University of Oxford. Her groundbreaking research program has focused on developing innovative vaccines against emerging and outbreak pathogens, and she is co-designer of the Oxford AstraZeneca COVID-19 vaccine. I'd like to say a special word of thanks to our alumni team led this evening by Melissa Byrne, who have put such preparation into tonight's event. The format for the evening includes a 40-minute conversation followed by a question and answer session, and we plan to wrap up by 8 p.m. Some housekeeping points for our audience tonight. Please feel free to submit questions throughout the conversation using Zoom's Q&A function at the bottom of your screen rather than the chat function. These questions will be sent to our moderator and panelists during the discussion, and we will try to get to as many as we can. Indeed, many of you have already submitted questions to us, and at the end of the event, we will provide a link to the HSE website and other sources of information which will cover off frequently asked questions around the vaccination rollout programme here in the Republic of Ireland. So thank you very much indeed then in advance to our panel, Cillian, Teresa and Karina, for your time. We were delighted you could each accept this invitation. We know it will be a really stimulating discussion. We truly appreciate it and we look forward to hearing from you. I will now hand over to you, Cillian, as our moderator for tonight's conversation.
Thanks, Professor Kelleher, for the kind introduction, kind words, and yeah, I'm really looking forward to speaking to Professor Butler and Professor Lambe this evening. So I might just dive right in. I'm conscious of our time. We're not going to be here all night. So Teresa, can I just ask you first to maybe give us some background on the adenovirus vector vaccine that AstraZeneca and you have developed over the last number of years? What has been used in previously? And then maybe we can lead into SARS coronavirus type 2 afterwards.
Sure, and a big thank you for hosting the event, and UCD alumni as well for inviting me. So the platform technology that we've worked on for about 15, 20 years is a ChAdOx platform technology in adenoviruses. And we'll all have been exposed to an adenovirus in our lifetime, and it causes an upper respiratory infection or a common cold. And what we've done is we've taken this cold virus and we've manipulated so it doesn't replicate anymore, it can't cause a cold anymore, but it's a really good platform technology for developing a vaccine. The extra step that we've also taken is, instead of it being a human adenovirus, because as I've said, we've all seen them, we'll have mounted an immune response to them, we've taken a chimp-derived adenovirus. So we've taken an adenovirus from a chimpanzee, we've crippled it so it doesn't replicate, and we use that almost as the carrier of the information that we need to make a vaccine against a particular disease. So for an example of a disease that we've previously used this vaccine technology for, and probably one that's quite pertinent, is the MERS coronavirus. And MERS coronavirus, or Middle Eastern Respiratory Syndrome, is a coronavirus that was discovered in 2012, and it can prove fatal. So what we did with MERS and what we did with SARS-CoV-2 and any other infectious disease is we looked at the virus and we tried to identify what part of the virus we thought, if we had a strong immune response against it, we might be able to mount a good immune response that would mediate protection from infection or disease. So most of these viruses have proteins on the outside of them, and these proteins mediate the entry into your body or the entry into your cells. And the protein we chose was the spike protein for MERS, and we developed a vaccine against MERS a number of years ago. We progressed it through GMP manufacture, and that's the manufacture of vaccine so it can go into humans. We progressed it through preclinical testing and did a small-scale phase 1 test with it. So we were relatively confident that we had a technology that we could use against the next disease X when it came. And much to my surprise, because I have to say that I'd banked on it being flu, it was a coronavirus, it was SARS-CoV-2. So what we did was we built on the information that we had against MERS and other coronaviruses, picked the protein on the outside of the virus, the protein that mediates it getting into your cells and gets us sick, stuck the information to make this tiny part of SARS-CoV-2 into our adenovirus platform, so that when we inject the adenovirus vector into our arm, it expresses this small part of the SARS-CoV-2 virus, the spike protein. Your body recognizes it as foreign. It mounts an immune response, so it generates antibodies and T cells. And then we trialed it through phase 1, 2, and 3 to try and see if we had an efficacious vaccine. I've probably spoken too much, so please interrupt me.
No, not at all. It's fascinating. I was just going to ask, so from memory, I think the first SARS-CoV-2 sequences were made available probably around the 10th or 12th of January. So I'm just curious, as to when did you guys start working on your vaccine?
So I started on the 11th. So yeah, my brother's not going to like this, especially as he went to UCD as well. I'm a protective older sister, and my brother was in Shanghai at the time. So any kind of outbreak pathogen that is going on in China, I tend to follow with a zealous interest of an overprotective sister. And there had been a lot of, well, not a lot, but there had been some conversation on Twitter about a virus that was spreading, but very little was known about it. And we had collaborators that work on virology and virus isolation. So as soon as the information was released, so it was the Friday night, but it came in past midnight. So I often say the 10th, but it was the 11th, that it arrived in my inbox. And I spent the weekend with a couple of other people designing this small part of the vaccine that we could slot in and then run with it.
Very good. And when did it first go into then preclinical or sort of phase 1 trials?
Yeah, so I'm probably going to fudge this slightly, Cillian, because most of the last year has been a bit of a blur. So we designed the vaccine over the weekend, ordered the genetic code, so the DNA that would basically code for this spike protein for our vaccine on the Monday. It took about 10 days for that genetic code to come back in. And then we did a bit of magic make and do in the lab, stuck it into our Chaddox-1, worked through weekends to get the first batch that would allow us to do the preclinical work. And I think it was in and around February 17th that we did our first preclinical. And obviously you have to do those tests before it can go into clinical testing. And it was, I do remember the first clinical test because that was a bit of a day. April 23rd, we had our first volunteer vaccinated.
Very good. And when you started that process, obviously you'd had some experience at MERS. Were you confident, or sort of how confident were you that it would be successful?
So I wasn't confident that it would be successful. I knew we could make a vaccine, but there are lots of infectious diseases out there that you can't make a vaccine against. So HIV is a great example. So I was pretty confident that we could make a vaccine, but whether it would be efficacious or not, we were hopeful, but we weren't sure. And I have to admit that when the Moderna and the Pfizer results came out later in the year, that was a big boost, because that meant we could make a vaccine and lots of people would be able to make a vaccine, and that we could have a lot of vaccines, which is what we need to make a real impact against this virus. So I wouldn't say that I was overly confident. I knew we could make a vaccine, but we needed to test it to see if it was going to be effective.
Yeah, of course, very good. Karina, I might come to you on that note about, I suppose, having the vaccines. There's been a lot of discussion, I guess, about NIAC, the National Immunisation Advisory Committee, in the last couple of days. So what, as chair of NIAC, I suppose, what is NIAC and what have you been looking at over the last 3-6 months as preclinical data has come out, as clinical data started to come out? What's the role of NIAC in this?
Well, NIAC, or the National Immunisation Advisory Group, we do nothing as exciting as Teresa in terms of making vaccines. And as I listen to her, it's just fascinating, because really, Teresa, what you've done is you've created a Trojan horse, isn't that right? With the shell of the virus that brings in the code that tells the cell, this is what you need to do to protect yourself, which is just so exciting. And to hear it go through, and the speed and the work that went in there, it's really the dedication, fantastic. I'm afraid we're much more mundane, but nonetheless an important role. So we are committing the College of Physicians. We are a body of health professionals from different disciplines, from different backgrounds. We have got, there's some doctors, there's nurses, there's pharmacists. We have representation from the regulatory bodies who attend the meetings. Cillian attends the meetings, is part of our committee. Representation from our surveillance scientists, and we have observers from our health service executive and the Department of Health. And our role has generally been a much quieter role until this year, where we look really at what the vaccines are that are authorized, that are approved, and look at where they fit into our program for vaccination in Ireland, and then make recommendations accordingly. So everyone will be most familiar with the childhood vaccination program, or for example, the flu vaccine. And it has got, very dynamic over recent years. Vaccination used to be, everyone thought it was just 3 vaccines and it was just stayed for a while. But there's been a lot of change and refining, and vaccination has really come to the forefront, but never as much as this year with SARS. And never has everything been subjected to so much scrutiny. As I was saying to Teresa just before we came online, is that she must be sitting back and wondering, she's probably never had people pore over each paper that has come out of the lab in such detail, even before it gets peer reviewed, because it's such an exciting work, but everybody wants to do the right thing and find the right niche for the vaccines that are available. So that's what we do. We look at the authorized vaccines, the upsides and the variations in them, and try and see how we can best use them within our national program to benefit both the individual, but also the population. And they're 2 different things. So we try and do that across the board.
And how confident were you, Karina, say 6 months ago or 9 months ago, that we would have a vaccine? Or the pace of development has been phenomenal, to think that where we've come from in the last 12 months. When did you realistically think we might have a vaccine, let alone the 3 that we have at the moment?
Well, I wasn't confident at all, because I suppose in my main background in terms of clinical research has been in HIV, and we've been through so many false starts from that point of view, and we've really relied on antivirals there. So I have to say, in the beginning, I thought this would be more a situation of getting a small molecule or an antiviral that would work. But things began to get a little bit optimistic for me, maybe a bit later than for Teresa. It was more around the autumn this year, and things really began to get exciting when suddenly around November, October, November time, you began to feel, yes, things are happening. But I think even the speed at the end caught us all by surprise, because even when things began to go into phase 3 trials, and that was very exciting, you felt it was going to take a time before we got enough events to be able to show that there was a difference. And then, and we were in the quieter phase at the end of the summer. But then, because the virus ramped up globally, really, then people recruited much faster, because again, that was a challenge. Would people enroll? Would they take novel technologies and get involved and volunteer for trials? And I have to say, so much credit is due to those early participants in all these trials, because it does take courage to put yourself forward and say, I put out my arm because this might really help people. So that was wonderful that they recruited very quickly. And yes, not everything happened quite as might be planned. Teresa will speak to that, because that's the real world. And then by the end of the year, we were talking about not 1 or 2, but 3 vaccines and a couple more in the pipeline. So now we are here. It's just amazing to watch the vaccine tracker and the numbers that have progressed along. A couple have fallen off the wagon, as it were, because they haven't worked out. That's what you'd expect. That also reminds you of the amount of work that people put in. And it's wonderful to have someone who's had success. But for that, there's lots of people who have really given of their time, their souls, their energy, and yet they mightn't enjoy this success. So I think success is due to everybody that has been involved in the field.
Absolutely. And Teresa, on that note, we are used to, I suppose, vaccine development taking a lot longer than 12 months. So why did it work why did it go so well this year? Obviously, people were concerned that, regulatory corners had been cut and that type of stuff. Obviously, none of that did happen, but maybe you can just give us some insight into how it progressed so quickly this year.
No, absolutely. And that's a really important piece to speak to as well, because I think people are a little concerned, especially when they hear experts like me saying it normally takes us 5-10 years to develop a vaccine. And there are different stages of developing a vaccine. So you initially have what I did over the weekend with my colleagues and friends, designing the vaccine. And then the next stage is the preclinical testing. And the stage after that is something I've touched on, the GMP manufacture. And that's a lot of money, moving from preclinical into GMP. So making a vaccine that can go into humans, it's a big investment. And then you start small because you want to make sure your vaccine is safe. And that is first and foremost the thing that we test in our clinical trials. It's the safety of vaccines. And after a positive readout from phase 1, you may move on to phase 2 and phase 3. And at each of those stages, there's a stop-go decision. And you also have to ramp up your manufacture. And that's a lot of money, it's a lot of effort, and it's a really difficult task. So you can see why a vaccine manufacture takes 5-10 years. So what we tried to do was a lot of things in parallel and at risk. And it wasn't at risk ever of the safety of our volunteers. And I have to commend those as well for coming forward. Completely agree that without those individuals, we wouldn't be where we are today. So we never did any shortcuts on volunteers, but what we did is we manufactured at risk. So we knew we had a platform technology. We were hopeful it would work. We had good signs from our MERS clinical trial, but we weren't guaranteed. So we sought investments and we sought partners, and that's when the AstraZeneca collaboration happened. And also we've got Serum Institute now, and they took a huge financial risk on this vaccine working or not. And they started to ramp up manufacture so that we would have vaccine if it did progress through phase 1, phase 2, phase 3. And the other thing that we did was we had a very open and free dialogue with regulators, ethics boards and safety boards as well. And those meetings that can occasionally take a couple of months to set up, we tried to have them more frequently. which gave us a lot more work, but was worth it in the end.
Very good. And Karina, on the issue of, say, the other alternative vaccines then, the likes of the mRNA, we haven't had mRNA vaccines in our schedule before. So we had I suppose the challenge, I guess, of communicating about the novel technology and explaining that to people. How challenging has that been in your role, say, from a NIAC perspective, from a communication perspective?
Well, I think, as I was saying to Teresa in describing her vaccine, we described it as the Trojan horse, where you've got the shell, which has been altered so that it can't make more infection, and it carries the blueprint or the architect's instruction as to how to make things. Well, in the RNA vaccines, it does it a little bit different. It's just the architect comes with the blueprint and sticks the blueprint in. They have to modify it a little bit to get it in there, but it's not quite the Trojan horse. But that's what it's like. It's taking the instructions, which is modified. It takes the RNA, has the instructions, manufactured it with the nanoparticle, the lipid nanoparticle, so that it can fuse and get into the cell, and then it's released and is able to instruct the cell how to make the proteins that are then expressed, and they instruct the body how to make the antibodies to fight infection. So I think it's about just basically trying to explain to people, answer the questions, what's going on, and then realizing what are the questions they have about it. So with the adenovirus vectors, is the adenovirus going to cause infection? So we're able to say in this one, no, it has been modified, so it doesn't. These are DNA and RNA. We're talking about genes, genetics. Is that going to integrate into my host genome? No, it's not. It doesn't have the power to do that. It doesn't have what's necessary to get in there. Is it going to hang around and alter me? No, it's not, because the RNA degrades very quickly. The RNA in the vaccines, it's hard to handle those vaccines even. You have to be very careful. Some of them need to be frozen. It doesn't stay around long in the body, but it stays around long enough to instruct the body how to stimulate the immune system. So I think we're all learning the ways as to what the questions are and how to find the answers, because we don't always know the answers right away, and then how to share that information. And I suppose events like this evening are about information sharing, because if everybody can get the information they need, or at least know where to find it, or realize that they have a good question, but we don't have the answer to that yet, but this is what we're doing about it, then I think that can give people the reassurance.
Absolutely. And in the context of, say, what we're seeing in the real world now as the vaccines start to be rolled out, have we seen anything, say, from a safety perspective that we would be concerned about, or are we very happy with the way things are going?
Well, I suppose in the first instance, right from the beginning, and Teresa will speak to this as well, even though the phase 1 studies are done in healthy volunteers and they're not looking for either efficacy at all or safety, you're observing from the very beginning. From the first time a vaccine goes into anyone, you're paying attention to safety. And through phase 1, phase 2, and then ultimately in the larger clinical trials. And in these clinical trials, it's been between 28,000 and 40,000 participants. Now, only half of those would have got the vaccine. The others would have had a control or dummy substance, and they're being monitored for safety. So after the clinical trial, you have a very good sense of what the short-term safety is. In these trials, the regulators wanted up to 2 months. Why did they want that time? Because actually with vaccines, we know that if there is an adverse reaction to vaccines, over 95% of them have onset within that 6-week period. It doesn't tell you everything about the vaccine, but what do we mean by safety in a vaccine? What we mean is that whatever residual risk there might be is far outweighed by the benefit, given the risk of the challenge that you're up against. So in this case, the challenge was coronavirus and what it was doing, and the risk that it was posing to everybody. And so, yes, we don't know everything in terms of the long-term, either the duration of immunity or the long-term possible unanticipated effects, but the risk of that will be very small, and the counterbalancing risk is the very real risk of the coronavirus infection. So that's what you're balancing up.
Yeah, absolutely. And Teresa, on that note about sort of the vaccines and the trials and the information we're gleaning, I suppose what work is ongoing now with, say, the AstraZeneca, with the Oxford vaccine? What trials are still ongoing? What what do we not yet know, and what additional information will we get from the trials that are still in progress?
So I was hopeful my life would get a little less busy after we had an efficacy readout, but that has not been the case whatsoever. And Karina touched on some of the really interesting questions that we're continuing to look at. So longevity. So April 23rd was the first time we put a vaccine in somebody's arm. So we're not even at a year yet, but we continue to look at the immune responses in these individuals and look at the antibodies. And antibodies are essentially the Y-shaped molecules that bind to the spike of the virus and prevent it getting it in. And those neutralizing antibodies are thought to be very important. So we're looking at the levels of those, whether they're still able to neutralize. And we're also doing some other studies in collaboration with other individuals looking at combination regimes. So that's a study that I'm not directly involved in, but within the department and with the vaccine task force, they're looking at RNA vaccines along with ChAdOx platforms to see if we can get the immune response induced with an RNA and a ChAdOx. We're also looking at the T-cell response. So the T-cell response, if a virus gets in and it's able to take over the machinery and you get poorly from it, a T-cell is really good at mopping up those cells and clearing it so you're less poorly, so it can result in a less severe disease. And we're also keeping an eye on all the variants and measuring the immune response towards the variants. So unfortunately, my workload hasn't got any lighter and I'm still, working silly hours.
Well, I can relate to that, so I sympathise.
I'm sure we all can.
And on the, there's been a lot of, I think there's a lot of people, I suppose, in recent weeks, I suppose, are becoming sort of amateur vaccinologists because of the media coverage has been incredible around all of this. There's been a lot of discussion about, say, the dosing interval between the first and the second dose, and why sometimes it might be better to wait 3 months as against a month. Similarly, obviously, AstraZeneca had the sort of the half dose, full dose regimen versus 2 full doses. And then the final question, I suppose, well, actually, maybe on those 2 issue questions first, what's happening sort of from an immunological perspective to make one regimen better than the other?
So that is keeping me busy as well. But we've just had a preprint put out today that helps kind of decipher those mysteries. So what we had seen with our first interim efficacy after a certain number of cases was an efficacy readout with the SDSD and then a different one with the LDSD. And I'm speaking in tongues slightly there. It was a full dose followed by full dose and then a half dose followed by a full dose. And we saw a higher efficacy with the half dose followed by the full dose. And the paper that we've just put up in preprint, and it is a preprint, so it needs to be peer reviewed, indicates that we're getting in and around 76% efficacy after 1 full dose, so out to 12 weeks. So that's quite high efficacy, in my opinion. And then after we give a boost, we get up to 82%. And it actually, what we've done is we've looked at the efficacy divided up by interval. And what I mean there in English is looking at the efficacy in regimes where you had a 4-week between your prime and your boost, an 8-week, a 12-week, and almost continuously. And what we've seen is the longer the interval, up to about 12 weeks, because that's where we've had a lot of the solid data, the efficacy tends to rise. And we also see an increase in the antibody titers with longer intervals. So that goes some way to explain the differences in the efficacies across the regimes, because the majority of individuals that got the half dose and the full dose had a longer interval. So we think a lot of it has to do with the interval between the prime and the boost.
Okay, and from an immunological perspective, is it fooling our immune system? Is it just giving our immune system more time in that intervening period to get better at recognizing the virus or the spike protein?
So the honest answer is I don't know yet, and we're working on it. So you would assume that what you're getting is higher affinity antibodies, so antibodies that bind stronger, and also that you're selecting for those antibodies better with the longer interval. But we need to prove that. So that's just a hypothesis at the moment.
Very good. And Karina, when you're looking at something that's evolving so quickly and with new data coming out on a certainly on a weekly, if
daily
not
basis.
Yeah, I feel like apologizing.
It's not daily basis. How I suppose how challenging is it to try and I suppose implement or bring? I appreciate you're not involved, say, in the operational rollout of vaccine, but just when you're trying to inform or advise government from an IAC perspective, how challenging is it to take in all of those different, the different regimens, the different dosing intervals, and to try and, I suppose, put that into a cohesive single message that people want to hear?
Well, it's very challenging. And the problem is that it also can give rise to confusion, because it's very easy if you can start with something that's black and white and you stick to it and you say, this is our plan and this is what it's going to be going forward. So that would make it easy, but that wouldn't necessarily be the best thing to do, because it is a very new situation and we're learning along the way. And what this has demanded of everybody, and is also demanding of the public at large, is, if you like, to have that adaptability and agility, as we say, to be able to embrace the information that's coming out and to go with it, in the sense that if we start out on a journey, if we're going out on a road, if we find, you know, that there's an obstruction or there's a tree down, we'll go around it. We're not just going to plow through it. Or if suddenly something happens and there's a clearer runway on one side, you'll move on to the new pathway. You won't stay on the old road if it's a bit bumpy. So we can't see straight down the tracks in this, except we know where we want to go and we're getting the information fed in all along the way. And every so often we have to make course corrections. Now, we can't guarantee that sometimes we won't have taken a dead end and may have to backtrack a little bit. But the thing that has been wonderful in this is it really has been building on the science that's there already. And everything so far that has really been coming out has been a positive. It's been one positive after the other. So you'll say, okay, we started with the 4 weeks and now maybe it's 12 weeks. And in our recommendation, we're saying 4 to 12 weeks. And maybe that's going to cause confusion. We don't know for certain yet that 12 weeks is going to be better, but maybe that will be. So, and particularly in the younger people, and it was mainly a younger cohort. Teresa will probably correct me if I'm saying wrong of her signs, but it was the younger cohort that had that longer dosing interval, and they did get, you know, quite higher antibodies, and that might last longer as well. So that might be a very reasonable to stretch out on that vaccine to the 12 weeks. It doesn't mean it will be the same for the mRNA vaccines. We might do differently there. But I'm saying we have to take the information on, be open to it, try and keep up with all the information that's coming out, liaise, because no one person can do this. So, for example, for us on NIAC, we had some of Teresa's colleagues speak to us over the weekend, share the information with us so that we could ask the questions, parse it. We had a European meeting on Monday, again, sharing of information, everyone looking and trying to get what's best and what fits. Because as I said, what we're aiming for really is we're aiming to protect the individual, but we're also aiming to protect the population and get the wider population through. And sometimes those things take slightly different modulations to do that. And here we're trying to do both at the same time.
Absolutely. And just on that note, I suppose over the last, I suppose, again, 24 hours, the decision around the use of the Oxford vaccine in the over 65s or over 70s has been discussed. I might just, if you would like an opportunity, just I suppose to explain that decision, because again, there's been a huge amount of interest in it. EMA went one way, or well, not sorry, that's unfair. EMA sort of licensed it for use in all ages.
The EMA licensed the AstraZeneca vaccine for, or the Oxford vaccine for all adult age groups, irrespective of age. And there's very good, at least looking at it as the outsider, the immunogenicity data in the earlier studies, in the phase 2 study, looked very good across the age cohorts. There really didn't seem to be any difference, and Teresa can give us any nuances she likes on that, but that looked very good across the age cohorts. So we had that, and we had the overall efficacy that has been accepted by the EMA at being around 60%. Now, that may be doing a little bit of discredit to the vaccine overall, and I think we may see more data coming out on that, but that's where we are. We know that it will do that. It's very good. It's much higher than we had hoped for. It's higher than the threshold that WHO had said that we would need for a vaccine to confer benefit. So it's above the bar. The thing that has been amazing is that we don't have one vaccine above the bar. We have now 3 vaccines above the bar. And so some of them are a little bit higher in this early protection, maybe others will have different, and it's about where do we get the right placement. Now, as it happens with the AstraZeneca vaccine, although we know that it has the same protective efficacy across the ages, just by chance on the ages that were enrolled, we don't have as many older people in those trials as in some of the others. So we don't have quite the same robustness of the data when we get up into the older age groups. So on the basis that they are the most at risk, we decided to use the vaccines that had the higher reported efficacy, at least for now, to try and deal with the individual protection. But we know that we need to use all of these vaccines. And again, there's that intriguing sort of window that maybe we are going to have a longer duration of protection, that we maybe won't need to come up with that second dose until 12 weeks. And that will be helpful in getting the vaccine to a wider population quicker. And the other thing that's interesting, and Teresa hasn't brought this out, it's very hard to be sitting here and talking about your data on your vaccine, but you know, sometimes it's good to see what we see from the other side. But there is intriguing analysis in terms of what the impact the AstraZeneca, and the others may follow, we just aren't there yet with them, has on the overall infection rates in a population, and how that will translate into to preventing transmission. Because right from the beginning, we said we had to focus on protecting against disease and morbidity, because that's all we knew at that time that the vaccines would do. And therefore, we were targeting the people who are most vulnerable, the people who would get sick, who would end up in hospital. If we knew that a vaccine was particularly good at stopping transmission, then it might be much more appropriate to target the people who don't get sick, who are out there spreading the virus around and targeting a younger population. So those are the kind of factors that you may end up seeing an array of vaccines, but each with a different niche that's best for them within the population. Teresa may want to come in on that.
No, actually, it's really good to hear somebody else speak to this point, because sometimes I get a bit tired of saying it. And to be honest, I'm just delighted that there's 3 vaccines available in Ireland and will be available to my parents as well. And you're absolutely right. The EMA have recommended for 18 years and over, and it's up to each country to decide the best placement of vaccines for their population as a whole. And you've pretty much summarized a lot of the data. So what we've seen is really good immunogenicity, and the immune responses, the antibody binding and the T cells in 18 to 55-year-olds and overall older adults. But we didn't enroll the older adults till a slightly later stage of the pandemic and also in our trial. So actually at the stage that we enrolled the older adults, the transmission had gone down, which was great, but not so great for an efficacy trial. And also older adults were cocooning, or they were at home rightly. So we're continuing to monitor them to look at the efficacy in those. And it's very likely to be similar to the 18 to 55-year-olds because the immunogenicity is similar. And we have the US trials as well, which is a larger population of older adults. So there's lots of data to come that will probably give you even more headaches, but it's all really important to consider.
And what we really want, Teresa, is we want you to come up with a threshold so that we know what that bar is. We want our level where we can say, yes, this correlates, because it's fascinating in the sense that we have this sense of, you know, our antibodies correlate with our level of, say, anti-spike antibody correlates with the neutralizing activity, and that seems to correlate with efficacy, but we really don't know. And we have that whole underlay of the T cells in the background as well, and how much of a role that plays in. So we just want you to give us something simple.
No, you're absolutely.
What's the correlative efficacy?
Yeah, no, you're absolutely right. And that is a huge body of work that we and many others are undertaking. So it's a correlate of protection. And basically we want to be able to delineate when does a vaccine get to the level that will mediate protection against severe and overt disease and also transmission, which I haven't touched on because you did such a good job of explaining. And they may be slightly different correlates. One bar might be higher for the transmission compared to protecting against severe and overt disease, but you're absolutely right. We really need to dig into that. And that will make future vaccine trials a lot easier to do because we'll be able to look at new vaccines and look and see if they reach this threshold, and you may get marketing authorization based on that threshold, as you can do with the influenza field. So it's a big piece of work, and I think a lot of us are already trying to answer your question.
And if I'm conscious now that I will need to go to our participants, because there's Zoom is telling me there's 99 plus questions which we're unlikely to get to before 8 o'clock, but hopefully it's a good sign of the level of interest. So I'll go to some of those, but just what I just wanted to ask, I suppose 2 quick questions from a personal perspective as a virologist. And I suppose the first one maybe to you, Teresa, from the spike protein. So you guys have used a spike, in simple terms, you guys used a spike protein that was there in January. That spike protein is now different. What impact do you think that is likely to have? Thankfully, at the moment, it doesn't appear to be having a significant impact, but are you already looking at a sort of an updated version, or what would that lead in look like?
That's kind of my bread and butter, is to be looking at new vaccines and developing the next disease X. So yeah, sorry, I'm being slightly facetious. We do need to continue to monitor the different variants, and we do need to measure whether they're having an impact against the vaccine immune responses and vaccine efficacy. And we are certainly keeping an eagle eye on all of these variants, and we will develop some preclinically and test them just from academic interest, but that will also give us an ability to push any forward if we think it's needed. So that's also giving me a lot of work to do between correlates, longevity, and new variant analysis. I've got enough on my plate.
It sounds like it. And then, Karina, just for you, if you had to put your money on something for the coming years, do you see this being an annual vaccination program for SARS-CoV-2, or what do you envisage?
Well, I never win the lottery, and I'm not a betting woman, but I think it may very well end up as something like that, just on what we know about coronaviruses in the past, and that maybe it won't be an annual, but it might be something that maybe every 2 years one needs to have a vaccine or a booster. I really think the way vaccinology has gone in the last year, I mean, I think this has just been a game changer for vaccination, and that these wizards might be able to concoct something even for flu that will be much more stable and give at least maybe 5 years, benefit, if not longer than that. So that would be really spectacular if they could.
Great. So, okay, look, I'm going to move to some of the questions from participants purely because I could chat all night because this is really interesting. So one question which I was thinking of myself, and again, in general terms, how is it, Teresa, that another company, you don't need to be sort of critical of another company necessarily, but how is it that in vaccinology that there is the possibility of a one-shot vaccine? That doesn't, obviously a lot of the vaccines we have at the moment obviously are 2 doses, and even in non-SARS-CoV-2 vaccinology, there's often sort of 2 doses, if not more. So how does a one-shot vaccine work, and can that give the same level of durability?
Sure, I'm going to take this back to my immunology roots because that's where I'm most comfortable. And really, it's also touching on something that Karina explained quite well. It's a correlate of protection. And it's inducing enough of an immune response that you are protected against disease or transmission, but certainly against severe disease. And if you can achieve that with a one-shot vaccine, that's great. But if you need 2 shots, so be it. Do the 2 shots.
Yeah, no, very good. Karina, have you thoughts on that?
Well, I think what people might mean by a one-shot vaccine is that it lasts a certain amount of time, but they may come back and then need another one shot in 6 months or in a year. I think in something like this, it's much more likely, again, going back to the immunology, we're essentially priming, but in order to get that durable immunity, I mean, there's just so many examples, even with fairly staid organisms like Haemophilus influenza or bacteria, where depending on the time, depending on the immune system, you often have to come back and give that booster. With those that you need to do when the immune system is a little bit more mature, you know, it's just, I don't know if a one shot will really last durable immunity, but we'll see. And
Karina, I might stick with you. There's a lot of interest, I suppose, on the, say, the allergy, the anaphylaxis story with the mRNAs. There was initial reports. What's our current sort of thinking on that?
Well, with any vaccine, there's always the chance of getting an anaphylactic reaction, but it's usually very, very rare. It's about 1 in 1.3 million. So this was an example where in clinical trials, nothing like that showed up, because actually the people enrolled in trials are generally healthy and they have a certain exclusions. And if someone has allergy, they mightn't be in the trial. And then when it went into post-clinical trials, and post-clinical trial is really a stage 4 because surveillance is still ongoing. No one has taken the eye off the ball. Everything has been watched very carefully. There were the first reports of allergic reactions. So was that a one-off, or was this something more? And then we got a few more reports, and it seemed, yes, the incidence was higher. And in fact, now it's estimated, initially thought, well, maybe it was as common as 1 in 100,000, but in fact, it's less than that now. And we're not quite sure what it is. They felt it might be due to some of the components of the vaccine. There's something in there called polyethylene glycol. Now, that's a very common substance that's in a whole range of things. I picked up my face cleanser the other day and it was actually in that as well. But a lot of us, it doesn't bother. If we eat it in foods, we put it on our skin. But for some people, they can get allergic reactions if they are injected with it. It's in some drugs, some preparations. Not sure, but that might be the reason. But the instance of allergic reactions is slightly higher. So what we say at the moment, if someone has had an anaphylactic reaction to a prior vaccine or to an injectable drug, not to food, not to anything they put on them, then they probably need to get specialist advice from an allergist or an immunologist at this time, and the mRNA vaccine might not be suitable. If someone has had an anaphylactic reaction to it, of course, they shouldn't have a second dose. So with the Moderna, there's also an increased risk of anaphylactic reaction, though at the moment it doesn't seem to be quite as high as with the Pfizer, a little bit less. And so far, we really don't have any signal in terms of the AstraZeneca vaccine in that. There have been some allergic reactions, but just at the background rate that you would expect, nothing really sticking out as far as I know as yet. And as I say, it's not definitively linked to the polyethylene glycol. There was concerns that it might be a cross-reactivity with another ingredient, but that other ingredient, polysorbate, is in lots and lots of vaccines that doesn't cause any problem at all generally. So people are still working that one out.
Great stuff, thank you. Teresa, a couple of questions.
I should say, sorry, Cillian, just for everyone, there is a frequently asked question up on the College of Physicians website regarding allergy, which the Allergy and Immunology Society of Ireland have very helpfully come together with us to put together to try and address those in a little bit more granular fashion.
Yeah, actually, that's a fantastic resource. I wish I had it as a medical student. I read it a couple of weeks ago. Teresa, there's a couple of questions on Sputnik. Obviously, you didn't work on Sputnik. How does that compare? What's it to say from an approach perspective, from a platform perspective, with say the Oxford vaccine?
So the Sputnik, I'm going to have to. It is another adenovirus technology. So as best to my recollection, it's an AdHu5, so that's a human adenovirus, serotype 5, and an AdHu26. So they're human adenoviruses, and it's used in a prime boost regime where you prime with one of the adenoviruses and then you boost with a different adenovirus. But a similar type of platform technology, so an adenovirus.
And I thought I heard something on the radio this morning suggesting that there might be an Oxford Sputnik combination approach. Is that?
Yep. So those studies are being predominantly led by our partners AstraZeneca. So I'm not sure what name that trial will come up with, but hopefully it's as good as Sputnik, but we'll see.
Very good. Sorry, so I'm just trying to feed. There's so many questions here, and it's trying to find one. So there's, yeah, we've answered the question about the whether it's likely to be an annual vaccine. So we're not certain about that, and we have a couple of questions about variants. So I suppose, yeah, just from the variant perspective, Teresa, I know we've touched on it, but just how challenging is that from the point of view of as you said, trying to, I suppose, keep an eye on it while obviously the first vaccine is still being rolled out, you're still collating data, still analysing data from that. How difficult is it to, I suppose, to horizon scan and have the underlying work in the background?
Yeah, no, that is difficult, and that is not my strength. I'm good at immunology and making vaccines, but not necessarily sequence analysis and virology. So we partner with others in that space, and also AstraZeneca are very helpful as well, and we're working closely with them.
Right, so I just, Karina, maybe if this one again. Yeah, go ahead.
No, I was just scanning through some of the questions as well. There were some quickly. People were asking about what about the over 65s to the 70s in terms of whether they would get the RNA vaccine or the AstraZeneca vaccine, and when would more details be available. The AstraZeneca have a large trial with those over 60 ongoing in the US that might be giving information this month. Therefore, we may have information by the end of the month, and that will inform us better. So that waiting for that to see. So that was just something I'd noticed going through there. Sorry, you were coming in with something, Cillian.
Yeah, no, I was just going to, again, there's been a couple of questions just around, I suppose, vaccine hesitancy, but the uptake, to my knowledge,
yes,
to date has been fantastic. So
the uptake so far, at least, has really been astounding in healthcare workers and in the nursing home residents. There have been very high uptake, and also in Department of Health, they've been conducting, I don't know if it was weekly or bi-weekly, but there have been frequent studies looking at vaccine acceptability, basically since about, I think, October or so, over time. And all that has happened is that the rates have gone up. As I often say, in one of the first kind of Q&As that I did for this, which was probably just before Christmas time, the whole questions were around being too rushed, too fast. How would we know we wouldn't take it? And it's like now it's totally flipped around and it's too slow. Please give it faster. We're ready. Here's my arm. So it has been amazing. So I think people have realized, well, first of all, unfortunately, and I suppose tragically, over this January has been a horrendous month in the country, and we have to recognize that. And the families that have been really touched by this, and it, you know, we sort of said at the beginning, it was kind of, you knew someone who knew someone, then you knew someone who'd had it, and now almost everybody has almost someone in their really quite close network who has been significantly affected. And with all, you know, it is a tragedy. I think lots of families are going through great difficulties related to this, and all we can do is extend our sympathy. But you know, it's happening to all of us, everybody. So, but it has turned around, and I think people realize the magnitude of the threat, and the fact that it's not just the threat to the virus. And we were talking about this earlier today, but we also realize that our response to the threat, which has been our lockdowns, our lack of education for our children, have also brought huge harms, if you like, to society as well. And so we sort of see this as the way out of those. So resolving not only the harm that's caused by the virus, but the harm that's caused by our necessary response to the virus. So vaccine is kind of what we're all hoping for. But meanwhile, we have to say, as of now, just because you're vaccinated doesn't mean you can drop your guard. Because even I think Teresa would say, we still have the potential for transmission as long as it's there. We still have to stick to social distancing, hand sanitizing, which is the one I think we all forget, and wearing masks properly, and doing those things and trying to keep our network of contacts to the essential. We all need to have contact with people, but to keep them to that minimum.
Yeah, no, absolutely, very important public health messages. Teresa, I might bring you back to a nice immunology one here for you. So I think it's from a colleague here in UCD. Is there any concern that immunity might develop to the adenovirus backbone, the Oxford vaccine, and might there be implications on that for future dosing?
Yeah, that's a good question. So it's basically, if you continue to use the adenovirus, will you induce an immune response against it and therefore not have a good immune response towards the spike protein? So essentially an immunity towards the Trojan horse instead of the spike protein. So what we have done is we have looked at the anti-Chadox neutralizing antibodies, so the antibodies that are induced against the Chadox, and also the antibodies that are induced against the spike protein. And there doesn't seem to be a very strong correlation. We've got other data that is looking at repeated vaccination, so individuals that we've vaccinated previously in other trials, and that we've invited back to be part of the COVID trial. And that data should be out soon.
Very good. Thanks a million. Karina, there's one here. How people will have heard terms, I suppose, such like efficacy and effectiveness used interchangeably and probably inappropriately in some situations. So does either of you want to come in on that, maybe just to explain to people what we mean by efficacy versus effectiveness?
Well, efficacy is really what we see in the clinical trial, in the control situation where everything has been done to protocol and we're able to see directly what the impact of the intervention is on the outcome. Now, when you roll it out into general use, you bring in a whole lot of other factors. You bring in the logistics of the distribution, you bring in the way it's been delivered in the community, you bring in a non-controlled population, because as I said, as I made reference to, for example, in selecting people for the trials, very often they may be relatively healthy. In many of these, they did have some other conditions, but stable conditions. They're generally, well characterized population. But once you start rolling the vaccines out generally, you've got a much more diverse population, and you have all the other things. If you've got a 2-dose vaccine, people mightn't always turn up for the second dose, or it might be a bit delayed, or nothing's controlled. And then you look and see how effective is your intervention, your vaccine in the population. So that's what we refer to as effectiveness, as distinct from the efficacy. At least that's my understanding of it. Teresa may want to elaborate.
No, I'm very happy to be on a panel where you're explaining everything so well.
Great stuff. So again, we're coming up to 8 o'clock. So just I try and finish up with maybe one or two short ones if I can. And I've lost the one I had now, but it's Karina, just in the context, sorry, yes, did you want to just touch on the pregnancy question? It's come up a couple of times, and obviously there's been coverage of it.
So basically, again, going back to the clinical trials, there were no pregnant women enrolled in these trials, at least no one who knew they were pregnant. As in real life situation, there were some people who were on the trials who did get pregnant, but they were small numbers. So these vaccines have not been studied in pregnant people. They're in the early animal studies, and this goes for all of them. There was nothing to suggest that there would be any particular problem in terms of, at least in fetal embryogenesis or development. And so when we don't have that data, obviously we can't recommend vaccines for that. But again, it comes to a risk-benefit analysis. And if there was no downside to COVID in a pregnant woman, you wouldn't even be thinking about it. But in this situation, unfortunately, we do know that there are downsides to COVID in pregnant women for those who can't protect themselves in other ways. There is a higher risk of prematurity. There is a higher risk of adverse outcome for the pregnant woman, particularly at the end of pregnancy. Now, those risks are small. It is a magnification of risk, but it is an increased risk. So if you can avoid the risks, if you can avoid COVID, if you can keep yourself safe, then that's the thing to do. But if for whatever reason you can't, it becomes a risk benefit. And there's nothing we know about these vaccines that suggest they would be harmful, and therefore it may be reasonable to get vaccinated. And that's where the balance comes in. So for example, for frontline healthcare workers who, as part of their everyday, we know the risks of COVID are higher in healthcare workers and they may be pregnant, it is reasonable for them to consider being vaccinated. If they've decided to do it, what we recommend is they don't do it in the first trimester. That's a general principle about all. And the only reason we say that you should complete your vaccination before you get to 32, 33 weeks is because, and we were talking about primarily the RNA vaccines first, we know that particularly with the second dose, you have a slightly higher risk of getting sort of fever, chills, feeling unwell. And you don't want that to be confusing things at a time when you might be approaching delivery and to interfere with the obstetric decisions at the time. So if you can't, if you are at persistent high risk of COVID, and the only way that you can reduce that risk is considering getting a vaccine, well, then that's what we would recommend, is go ahead and get the vaccine, taking that into account. And like that, there is on the College of Physicians website, there is a frequently asked question, and there's kind of a shared decision tree to help people think about the pros and cons. There's no right answer or wrong answer here. And it does come down to the individual risk and making an informed decision as to what they wish to do.
Great stuff. Listen, thanks a million, Karina, for that. So listen, I'm conscious we're out of time now, so thank you to all the participants. Apologies to anyone whose question I didn't get to, which is the vast majority of you. So you'll have to give me negative feedback as a moderator in the future. It's been a real privilege. I just want to thank Professor Karina Butler and Professor Teresa Lambe. It's been a great chat. I've learned a huge amount, and hopefully so have all of you listening to us and watching us. I want to thank the UCD Alumni Association for setting up this webinar and this whole series. Hopefully that people are finding them useful and educational. So thank you all very much, and have a good evening and stay safe.
Thank you.
Thank you.
Magnificent. Thank you.